Hi, people.
Uh,
I hope you can see and hear us. We're going over my review notes for cell biology and life science in a nutshell. Um, so I believe this is the ninth video, if I counted right. But we were doing parts of the cell. Currently, we're studying about the cytoskeleton, which controls the shape and the way the cell you know, um, controls its shape throughout the cell cycle, and as the interaction between the 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 cytoskeleton and the different parts of the cell and the cell membrane, etc., and the proteins embedded in the cell membrane, all that stuff. So, Ruben, can you continue reading? We're over. So eukaryotic cells.
and eukaryotes, the
Not coyotes. In eukaryotes, the cytoskeletal matrix. Go ahead.
eukaryotes
Eukaryotes.
and eukaryotes, the
Cytoskeletal.
cytoskeletal matrix is a dynamic structure composed of three main proteins, which are capable of rapid growth or disassembly dependent on the cell's requirements at a certain period of time. The structure, function, and dynamic behavior of the cytoskeleton can be very different depending in, on organisms and cell type. Every Even with in one cell, the cytoskeleton can change through association with other proteins, although the cytoskeletal system of different organisms are composed of similar proteins. A multitude of functions can be performed by the cytoskeleton. Its primary function would be to give the cell its shape and mechanical resistance to deformation. And through association with extracellular connective tissue and other cells, it stabilizes entire tissues. The cytoskeleton can also contract, thereby deforming the cell and the cell's environment and allowing cells to migrate more ever. Moreover, it is involved in many cell signaling pathways and the uptake of
Extra.
extracellular material. Endocytosis segregates chromosomes during cellular division, is involved in
Kinesis.
cytokinesis, the division of a mother cell into two daughter cells, provides a scaffold to organize the contents of the cell in space and for intercellular transport. For example, the movement of vessels and organelles within the cell, and can be a template for the construction of a cell wall. Furthermore, it forms specialized structures such as flagella, cilia, lamellipodia, and protosomes.
Great job. So here in the picture, we have the stainage of cytoskeleton of the eukaryotic cell. Actin filaments are shown in red. They are more thinner, and you can see them more on the outside. Microtubules are more thicker. They are beta tubulins. In this case, the components of the microtubules, and they could be either alpha or beta. So here they're in green, so they're more thicker and towards the center. This is a beautiful picture. So, a main component in the cytoskeleton the true function of contraction is the microfilament. It says, we're gonna start learning a little more about these, um, which make up the cytoskeleton. Microfilaments are composed of the most abundant cellular protein known as actin. During contraction of muscles, especially so within each muscle cell, myosin molecular motors are used, collectively exerting forces on parallel actin filaments. Increases in calcium in the cytosol allows muscle contraction to begin with the help of the two proteins tropomyosin and troponin. Tropomyosin inhibits interaction between actin and myosin, while troponin senses the increase in calcium and releases inhibition. This action contracts the cell. So, through the synchronous process in many muscle cells, the entire muscle is able to move and contract. So, these are showing the muscle cells in green. Microfilaments are composed of linear polymers of G actin proteins and generate force as they pull and move and tug. Um, the G-actin monomer combines to form a polymer which continues to form the microfilament actin filament. These subunits assemble into two chains intertwining into what is called F-actin chains. Myosin motoring along F-actin filaments generates forces of contraction called actomyosin in the so-called actomyosin fibers, both in muscle cells and non-muscle cell types, where there's movement. Actin structures are controlled by a row family of small GTP binding proteins, such as Rho itself, for contractile actomyosin filaments, and also known as stress fibers. So they're helper proteins. Go.
Microtubules vibrate within neutrons in the brain, which suggests that brain waves come from deeper microtubule vibrations. Each cytoskeletal filament type is formed by polymerization of a distinct type of protein subunit and has its own characteristic shape and intercellular distribution. Microfilaments are polymers of the protein actin and are 7 nm
Hold on 1 second.
in diameter.
Okay, so sorry, microfilament. So it's
Are polymers?
yeah. So microtubules, um, and then microfilaments are 7 nanometers. They're small, tiny compared to microtubules, which are like 25 nanometers. Okay, microtubules are composed of tubulin and are 25. Intermediate are composed of various proteins depending on type of cell in which they're found. They're 8 to 12. Okay, so that's comparing their sizes. The cytoskeleton provides the cell with structure and shape, and by excluding macromolecules from some of the cytosol, cytoskeletal elements interact extensively and intimately with the cellular membranes in doing this. That's how they control what goes inside, the cell, and what does it. So in the pictures we have microtubules in gel fixation of the cell. This left one, cross-section diagram through cilium, showing 9-2 arrangement of microtubules. So that's the way the tubes are arranged, the subunits. And then, so microtubules have a very dynamic behavior, binding GDP for polymerization. So they need energy to do this. They're commonly organized by the centrosome. So this is a cross section, so showing how the cylinders look like from the side of the microtubules. They're pretty wide in comparison to like microfilaments. Say bye,
Bye
Ruby.
bye.
See you in the next time.